Alright, as promised, now that we understand our somatic cells and gamete cells, let's look at cell replication. Somatic cell replication is called mitosis, and mitosis can be broken down into four phases: prophase, metaphase, anaphase, and telophase. Let's go ahead and look at these phases in a little bit more detail. Let's start with prophase. So prophase is when DNA duplication has resulted in chromosome duplication. DNA duplication is something we reviewed last week. So DNA has duplicated enough times where these chromosomes are actually now in this duplicated form right in here. And bring in another character. The character is called centrosomes. These are little organelles that are going to come in really handy in just a little bit. These little organelles position themselves on the sides of the cell. So in prophase, we have DNA duplication that has resulted in chromosome duplication, and the centrosomes have positioned themselves on either side of the nucleus. The last thing that happens in prophase is this membrane, this nuclear membrane, starts to dissipate. This sets us up for metaphase. Metaphase is when the duplicated chromosomes align themselves in the center of the cell, and these centrosomes, which I told you are going to come into play later, basically attach themselves to these chromosomes, prepared to separate these duplicated chromosomes. Little grappling hooks basically coming in and grabbing a hold of these chromosomes. And this sets us up for anaphase. So going back really quickly, in metaphase we have these duplicated chromosomes. So remember, this is somatic cells, so there are 46 chromosomes. They're duplicated right now, so 46 duplicated chromosomes. And in anaphase, these duplicated chromosomes are pulled apart by the centrosomes. And the centrosomes move these duplicated chromosomes to each end of the cell. So here now on one end of the cell we have 46 regular chromosomes, and on this side we have 46 regular chromosomes. Moving on to telophase. Telophase is the final stage of mitosis. This is where we start to see components of a new cell begin to appear. So we start to see all those little organelles, we start to see all the little functions of a cell, and we start to see two new nuclei start to appear. So this nuclear membrane forms around this side as well as this side. We basically have a Siamese twin type cell. So there is no cell division yet. This is just the last phase in mitosis. The actual part of the cell going from one cell with two identical parts to two cells is called cytokinesis. Cytokinesis is the stage after mitosis, and this is when the cell divides to form two identical cells. Alright, so that's somatic cell division. Let's go ahead and take a look at gamete cell division or gamete production, a process we call meiosis. Now, since the last process was way too simple, I'm sure you'll all have it all down, meiosis is a little bit more complicated. It goes through the similar phases. It goes through prophase, metaphase, anaphase, and telophase, but it has to go through these phases again. So we have meiosis 1 and meiosis 2, and that's because of something that happens right around here. Here, let's talk about it a little bit closer. So meiosis in prophase 1 is really similar to mitosis, except for one key distinguishing feature, is that these duplicated chromosomes kind of align with that homologous other. So the one from your mom and one from your dad, they kind of align and they get tangled up. And this tangled up is called crossover. So in prophase, similar to mitosis, except these duplicated chromosomes align with the homologous other and they get tangled up. Now once they're tangled up in this part we call crossover, they exchange genetic material. This exchange of genetic material is called recombination. Just a little heads up, crossover and recombination is a question a lot of people get wrong on. Let's say, if it was to pop up on an exam. So while they're tangled up in this prophase, they exchange genetic material, and this is called recombination. So now we have two duplicate chromosomes with a completely new combination of genetic material. Let's move on to metaphase 1. It's like mitosis. Chromosomes align at the center of the cell. Centrosomes attach themselves to these chromosomes. And in anaphase, they're pulled apart, except again, one key distinguishing feature. They're pulled apart as pairs. These duplicate chromosomes are not separated, which means that we have 23 on this side that are duplicated with this new genetic material and 23 on this side that are duplicated with this new genetic material. Unlike mitosis, where we had 46 and 46, here we have 23 and 23. So let's go ahead and move on to telophase, where we see the membranes form around these chromosomes, new cell parts form, and cytokinesis, where we now have two cells with 23 unique chromosomes, but these chromosomes are still in this duplicated form. So what do we do now? We need to get these duplicated chromosomes into single chromosomes. That's why we have prophase 2 or meiosis 2. So in meiosis 2 and prophase 2, there is no DNA replication. The aim is just to separate these duplicated chromosomes. And so we start to see similar patterns appear. Metaphase, they line up at the center of the cell. Anaphase, they get pulled apart. Now we're getting pulled apart. And on this side we have 23, and on this side we have 23 because they're no longer in this duplicated form. And in telophase 2, we have the same thing, features forming on one side, features forming on another side. And in cytokinesis, we now have cell division on this one cell, cell division on another cell. So we basically went from one cell to 4 cells with 23 new chromosomes, or diploid cells, or gamete cells. And this is basically the process of mitosis and meiosis. I know your book explains this in some detail. I thought it might be helpful to go over it one more time, and hopefully you're able to understand this a little bit better. All right, let's go ahead now and look at how RNA or DNA creates you.